this house alive. Gonna set the whole goddamn place on fire.
Sorry for
Oh, it's
being
all
so
good.
late. Ah,
How are you doing
We
tonight?
are super excited to have you, so no problem.
They say the best things in life are waiting for, I believe. So
Worth waiting for.
yeah. So,
yeah, which way is that? Waiting
Yeah.
for you or you waiting for me?
it's all good. Thank you very much for joining us tonight. This is one of those ones where, to be honest, like, we were kind of threw an email out there, hoping but half expecting to not even get acknowledged. And then when we heard back from you guys, like with like not very long, like within a couple hours, it was like, OMG, OMG, OMG. And I'm texting with Fluff and like, oh, look, they're getting back to us. And so thank you very much for your time. This is awesome. Let's get right into it, I guess. So one of the things that I've kind of always wondered is why soil biology? Of all the fields that you could have gone into out there, why this one?
Gosh, I think trying to think back that many years ago. Why this one? I really liked microbiology when I took a class in college, the last quarter of my senior year, of course. I really enjoyed those organisms and counting bacteria and figuring out who they are and what they're doing. And it was interesting to me that a lot of people had no idea of what these organisms did in the soil. When I went to Texas A&M for my master's degree in marine microbiology, I was working with a lot of the marine microorganisms. And so I was looking at what are the bacteria and other organisms present in the digestive systems of the oysters, and showed that it was really critically important for these oysters to have the right kind of bacteria and other organisms in their digestive system, or they would not grow. And so that was really fascinating. My husband and I got married when I was doing our master's degrees. And when we decided that we both wanted to do our PhDs, we were looking for universities that would hire both of us for a research assistant position where we would do some teaching. We would be involved in some research that was going on. And the only place that gave both my husband and myself research assistantship was Colorado State University. So that's where we went. And I remember walking into the laboratory and saying, so what is it that you really do in this lab? And it turned out they worked on soil microorganisms. So my PhD work centered on fungi. And the first thing my major professor had me do when we kind of figured out what my research project was going to be was to go around to all of the other professors involved with soil. So like soil chemistry, growing things in greenhouses, horticulture, where they're dealing with trees instead of grass, agriculture of all kinds, and went to all of these different professors. And gosh, there must have been something like 15 of those professors. And I explained my research work that I wanted to do on fungi. I wanted to understand why fungi were present in the soil. What else did they do other than diseases and pest organisms? They had to be there for a reason. And when I explained that and got done talking about what I was going to do for my PhD to a person, each one of those people said, you better not do that, because you will never get a job working with organisms in the soil. Those organisms are just there. They don't do anything important. They're just there. And so how are you going to get a job with, but when you're an expert about something that doesn't matter? And it just amazed me to listen to that here's Mother Nature, and she has been making certain that bacteria have existed on this planet for the last 4 billion years. Those are organisms that do nothing? I don't think so. Just because we haven't figured out what the something is doesn't mean they are worthless. Well, fungi were even more than worthless. Absolutely nobody studied mycology except for taxonomists. That kind of attitude was pretty amazing to me too. So I kind of walked away from all of those chats with the existing soil people and kind of said to myself, I'm going to show them that there are organisms in the soil, bacteria and fungi and protozoa and nematodes and microarthropods and earthworms and incatraids and all of these creatures in the soil. And they were there for a reason. Nature wouldn't keep organisms that do nothing, just suck up, all of the energy and waste it all, and, spending their whole entire life happy and not doing anything important. She just wouldn't have it that way. So it was up to me to start to really understand what all of these organisms did in the soil. And so my major professor, Dr. Donald Klein, had me work on this project where we were looking at how do you measure, how do you test to make certain that the organisms are doing something. What's the something? So when I worked for my postdoc at Colorado State University, we expanded more information about showing how much nitrogen is turned back into a form of nitrogen that your plant can take up. When that nitrogen is out there in the rocks and the pebbles and the boulders and parent material, all of that stuff, that's not available to plants. Plants have no enzymes to break down the silica bilayer that is what makes up all of these rocks and mineral forms. So how did nature get those nutrients from the rocks into a form that the plant could take up? And so we worked on that process, and we showed very clearly where we added bacteria and fungi, your plant would do much better than just having bacteria. You had to have also, at the same time, you had to have protozoa, and you needed nematodes. And so nutrient cycling was really something done by the whole community. It's kind of like if you think of human goods and services, it's never just one person that's going to be involved in doing things. It's a whole company. They've got to have accountants and lawyers and all of these subsidiaries. So that's what the food web is, a combination of all these organisms. Well, as soon as we got done looking at nutrient cycling, well, what else might these organisms do in soil? Well, they could differentiate, they could make certain that when you're dealing with very early successional stages that need nitrate as a form of nitrogen, that the organisms that were present there were those organisms that would produce nitrate. So you didn't need to put out a nitrate fertilizer if you're trying to grow those what we call weedy plants or early successional herbs and maybe vegetables, things like that. As the bacteria are replaced, or you get fungi coming along in and start to grow and have enough food, then the fungi change the form of nitrogen that's present in that soil and utilizes the nitrates and produces ammonium instead of nitrate. And when you do that, you select against the weeds and the pest organisms, and you select for those forms of nitrogen that plants can take up, soluble nutrients, and start to promote those other organisms that are, and your plant, so that the plant would be taking up the right form of nitrogen, and the pH in the soil would remain what those plants required. So there's all these roles and functions of what organisms do in the soil. And to have been part of that change in understanding of the importance of soil and the nutrients present in that soil, it was a delight to me to be able to participate in all of that.
And we thank you all. I mean, it's
That's
amazing.
incredible.
It's hard for me to kind of even understand. I've had so many questions to ask, and now I don't even know. No, I was there was actually a couple of questions in chat and some other things that you know, our thoughts. A lot of us are kind of indoor gardeners, right? And we always think of what I was saying is we're rapidly destroying this earth, and we're looking at options to is the soil food web something that can be recreated, established, and held on to indoors? Is this something that we can function consistently?
Yeah, I think we can. It should be possible. The question comes where will you be able to keep the huge diversity of these organisms? If you're just in a teeny tiny, a greenhouse, 10 foot by 20 foot or something like that, you aren't going to be able to have all the variety and habitats present that will promote the growth, the continued existence of all of the different bacteria and fungi and protozoa and nematodes and micro earthworms, all of that stuff. So we would have to have acres and acres and acres of greenhouse in order to make certain that we've gotten all of the diversity that we need. And then think about all the different places in the world. They have different biomes, and we would have to have a huge, greenhouse operation for each part of the soil.
Yeah, it's like an Amazon warehouse full of soil for every specific area.
Every different zone, basically.
Yeah, pretty much.
Just to grow enough food for everybody on the planet, we would have to take over all of the current agricultural areas and turn it into the greenhouse.
Yeah, we'd almost have to put walls over the top of our fields to keep us from intruding any further.
Just wire everything so that when you walk along and you finally get through to the actual plants, then you get to go.
There you go. There you go. No, it's fantastic. There was other things. How do we go about like rebuilding some of these broken areas, these destroyed soils, things like this? How can we do those kind of things using your practices?
Well, we're going to want to collect starting materials to make our compost. And the reason why you have to do composting properly is that you kill most of the diseases and the pests when you understand what are the conditions that the bad guys, if you will, if I can shorten that terminology to bad guys and good guys. We know the conditions that select against the bad guys and for the good guys. And so we want to have make certain we go through those conditions. High temperature, achieved very rapidly. Most of the disease-causing, pest-causing organisms, pest organisms, are not capable of taking that rapid an increase in temperature. And then especially if you have it over a 3-day period of time where you get up to the high temperatures and maintain that high temperature in the compost, you've pretty much done away with all of the diseases and the pests. Certainly their eggs, so the adults, if they're in your pile, whatever, We're going to deal with those organisms. We want to make certain that at no time does your compost pile become anaerobic, because anaerobic conditions help select for the disease causers. Most of the disease causing organisms are in that early successional stage, and they grow very rapidly on an unprotected host. So we also want to hold on, I want to talk to my husband real quick. Russ, could you turn that down, please? So, so getting the right sets of microorganisms growing in a positive, a good environment, we can make certain that disease-causing organisms, pest and problem organisms cannot survive that composting process. So, we have a variety of ways of assessing that. The easiest way to assess whether you've got the beneficials or the not so wonderful species is to just get a microscope, not a real expensive one, something around, maybe $300 to $400. Nice camera that goes on it so you can take pictures. And when you can't identify something, you don't know if this is a good organism or a bad organism, you send it in to us. As soon as you start sending stuff into us, we're going to suggest to you that maybe you should take the foundation courses, because that's where we start getting people to understand all of the things that benefit the growth of your plant, not just nutrient uptake, not just getting rid of the competition of the weedy species, getting rid of the diseases and the pests. We also want to build structure within the soil so your root systems can grow as deep down into the soil as they possibly can go. When we've gone out on people's property and like they've had a dead oak tree that dies, okay, so let us come along and dig out how deep the root system of your plant was going. This was a oak tree that was 400 years old. And how deep down did the root system go? And we got opinions of all these arboricultural people, and they said, well, typically roots of trees don't go down more than 3 or 4 feet. And I kind of went, you better be here when we get to the bottom of the root system of this plant, because I think we might surprise you. 300 feet later, we finally were not finding root systems, roots from that plant. Most people who follow or fell into the trap of the Green Revolution believe that root systems of trees or plants, or like how far down do grasses go? How far down does your corn plant go? How far down are the root systems? How deep is that going? Well, again, the answer you're going to get is that those root systems don't go more than maybe 3 or 4 inches down, and then they go sideways. And what you have to realize is the roots going sideways means that they couldn't get through the anaerobic layer, that if the roots grow in there, they're going to get killed. They're going to get killed by the diseases and the pests that are promoted under those conditions. Low oxygen concentration, so it's anaerobic. Guess which organisms are going to be most selected for? Those things that will kill your plant, and they do a very good job of killing your plant. We need to promote the health of both the soil and health of the plants by getting the root systems to go down so they're not fighting with each other for a very limited amount of nutrients. So, you want to make some guesses about how deep down the root systems of corn plants go? 15 to 25 feet. So
I was way off. I was gonna say like 2 or 3. I was giving it a lot, but
yep. So, and these are all things you can do yourself to take a look at how far down are the root systems actually going. It's not like you have to believe me. Start taking a look by digging down on a few of your corn plants after you harvest and find out how far down those root systems actually go.
I did that with a small section this year. Actually, we had a lot of flooding, and I took and put some tomatoes out and kind of got them out way too wet, and did not plant them deep enough so that they stayed in that kind of, like you said, the wet anaerobic. And I bet they only had 3-4 inch deep roots, but they spread really wide, but it could barely even hold the plant in the soil because they were so shallow. And they were not productive in any meaningful manner.
They're, the roots that are going out sideways are just kind of holding on for dear, life, so that at least they don't blow away when the wind blows or when there's another, but yeah, you need to get the biology that builds structure in the soil. And so in that list of organisms in the soil, bacteria, fungi, protozoa, nematodes, microarthropods, earthworms, who are the most important organisms in building structure in your soil so that they're opening up, getting rid of all of that compaction, opening that soil up to oxygen moving back in, and building the structures that hold water so you don't have to irrigate in the late part of the summer. So we've grown grapevines for the grape community in Australia, as well as table grapes, on absolutely no water coming from the big, where they're storing the water on the top of the ground. The name of that, yeah, I love it. When I get older, I don't remember all the words anymore. So coming, they don't need to be taking that water away. They can grow more grapevines. They can grow more of any plant that they want if you're storing water in the soil.
Yeah, I definitely found that. I don't like to usually cover crop my garden because I like the look of, my main plants and then my mulch. And I definitely have that problem late season where I don't have that great water retention that needs to be there. And I end up watering way too much to keep myself productive or even be productive at all because of, that's usually when you're really trying to push good late stage, fruit production and things. So,
yep. And the time the microorganisms are going to build those structures for you so the roots can go down 10, 20 feet, then you've missed that opening, that time where you can make the organisms build that structure and then fill up those pores with water so that the roots grow down and get the water that they need without you having to constantly go out and irrigate. So, in California, we were growing corn and tomatoes and okra and all kinds of things, and never, and barely watering at all. Maybe once a week we would give them a bit of a spritz, just to, especially if it was especially dry. But really, they could have held on all by themselves, I suspect. Another experiment that we need to do.
Yeah, it's very interesting. Somebody in there was talking about some like cold composting. And that was one that definitely does interest me because we all know that everything will break down if you just leave it out there long enough, right? It'll just break down and turn into your soil and it'll be fine.
But it won't have the organisms that you want to promote by getting it into the, and you're not going to continue putting out compost forever. You'll get all the organisms in the soil. And once you've got the diversity up to where you need it to be, it maintains itself.
Yes. Right. And that's and what are some of the actual drawbacks to doing it that lazy way? Because things like you were saying earlier, is the bacteria and the bad bacteria and things, they can't live through that heat, that rapid, when you cold compost, they absolutely can. So, what are the drawbacks to doing it that way?
You have to have some organism present that is going to process all of the soil in your pile So worm bins are great. You don't have to reach high temperatures, because as long as all of those earthworms are rising up to where you put on the layer of food, and they will chew through all of that. Anything that touches the outside of an earthworm, or anything that goes through its digestive system and out the other end as um, a earthworm poop, they have dealt with all of the pathogens and the pests and the problem organisms typically. So there are other ways. It just takes a very long time. You know how patient human beings are. Sorry, wrong, not true. So we don't have the patience in many instances. And so you have to use the thermal approach. If you've forgotten to put any compost together, you only need to 21 days. If you know what you're doing, you can go from just starting material to finished product in 21 days. Well, other people, it takes them 28, or it takes, another 10 days or something. Close enough. When we get to any of the alternative methods that are not going to raise the high temperature, then it may take you a year or 2 years before that material is ready to be put out in the field. So I don't know about you, but I don't have enough land in most of the instances that we're working with. It's perfectly fine for somebody who's just got a 20-foot long by 4-foot wide bed, and that's their garden. You can make enough piles to keep putting in those organisms every year. So
Even then,
kind
I find it slightly hard with a worm bin to keep up with the needs, even in just my little indoor garden. I keep up with the needs of my home composting, but I'd never feel like it's done enough to use properly when I really want it. Because like you said, it takes forever for it to properly, get down to that grade that I want to use.
Yep, and you know that it has reached the place that you want, that you are safe to use it, when you look at it with your microscope and you see the bacteria and the fungi and the protozoa and the nematodes. Those are the really critical 4 that you need to look into, and make sure that you've got at least 135 micrograms of fungal biomass, 135 micrograms of bacterial biomass. You've got to have at least 10,000 flagellates and amoebae per gram of soil. And with the nematodes, you need somewhere around 100 nematodes. And some of those should be bacterial feeders, some should be fungal feeders, and some of them should be predatory nematodes that go out and eat other nematodes. Preferably they're eating your root feeding nematodes for you.
Is That's actually been an interesting one that I've really been wanting to kind of get into research and use more in my garden, was nematodes. Is there a way to collect yourself? If I wanted to inoculate my garden, to collect, inspect, and inoculate.
Yeah, well, the easiest way to get a concentrated amount of the nematodes or the microarthropods, depending on if you want to add that group to the system, is to use a Tellgren funnel. And we go through how you make a Tellgren funnel, how you set it up to extract the microarthropods, or Behrman funnel if you're looking at the nematodes. And you can very quickly concentrate hundreds of thousands of those nematodes, and then add that into a compost that, for some reason, the nematodes decided all to leave. Or it got too hot, it got too cold, there wasn't enough food for it, and they had elsewhere looking for things to eat, just like your earthworms do too. If something's wrong in that box, they'll let you know, because they'll all be crawling up the side and out across the floor, and that's really fun and exciting to try to put them all back.
Yeah, it's you know, I wonder these. It's really interesting to do these collections and things. I think that's one of the things a lot of us on this panel really like to do is the collection of all those, and then we all like to play sorcerer in our garden is what it really is. So
A little bit of this, a little bit of that. Let's try some of, yeah, yep. I think Cariboo Hart, you were trying to ask a question or something?
Yeah, I was talking while muted. It's common around here. We've got a question in the chat here. What's your thoughts on the quality of municipal compost?
It's not compost. And they shouldn't be allowed to call it compost in most instances. They haven't killed the disease-causing organisms. They don't have the predators in the system. There is no way you could rebuild structure using those materials. It's got toxins, it's got heavy metals. There's just it's a world of woe when it gets called compost, because it isn't. If you go back in time and you look at what happened when we started getting so many people concentrated in cities, what do you do with all their wastes? And so we started taking all that waste material, digging big holes in the ground just outside the city, and taking all that waste material and putting it into that landfill. Well, pretty rapidly, the city grew around the dump site and on past it, and then they filled up the landfill. So now they're looking for someplace else to buy the land and put in another landfill. And of course, all the people living around that area said, "Oh no, oh no, no, landfills stink, and because they smell really bad, that will lower the price tag on the houses when we want to sell them." So, where does the stink come from when we're dealing with putrefied organic matter, really is what it should actually be called? It's because under anaerobic conditions, the chemicals that are made in that compost are very toxic. So, ammonia, for example, how long can you hold a bottle of ammonia to your nose and breathe it? You would be doing irreparable damage to the inside of your body if you did that. Well, that's what's going on with all of the beneficial organisms in that putrefied organic matter. So they, with the landfills, they started to take all of the organic material out and required that anything that was plant material or organic material had to be composted. And so it's really, they're trying to reduce the volume that's going into the landfill every year. And one way to do that is put big windrows, 15 feet high, 24 feet high, 15, the width corresponding, and maybe they turn it 5 times while they're sitting on the property, but they go anaerobic within about 2 days, 2-3 days. Anaerobic as all get out. It stinks to high heaven. It's an indicator we always talk with our students about, is one good way to determine if this is good compost is that if you pick some of them from something from the middle of the pile and bring it up to your face, and you start, as it starts getting closer to you, you're going, "Oh, rotten egg smell mixed with I don't know what that is. Oh, ammonia, mixed with a little sulfur, mixed with a little this, a little that," and it's hard. Well, we all know what it smells like, so I shouldn't go on and on about that. So, you have to make real compost where you don't allow the toxic materials to be made. If you go anaerobic, it's going to be killing all the good guys. So, what do you do then when you realize that you're making something really very harmful? And indeed, when I first started working with compost, I'd go out to folks and say, "So, we can take your green starting materials and we can turn it into compost and we can show you how to make the piles." And they were like, "Oh no, compost stinks. I don't want anything to do with compost. Wait, where does compost stinks come from? Well, it's putrefied organic matter when you don't turn when you need to turn and get the oxygen back into the pile. Then it'll go anaerobic on you and it will kill the good guys and it will promote the bad guys. So that was the next thing I had to do in my career, was figure out how to make good compost with all of that vast diversity in it. How do you do that? What are the steps? And if you go back to Sir Albert Howard, who is the person who brought this kind of composting back from India, where he served as a government official for the Queen. And when he came back to England, he taught other people how to make thermal compost. And it was, people were just astonished. They were amazed. Well, why didn't we keep that kind of composting? Well, because we had this vast amount of organic material that we had to deal with. And the only thing they could think of to do was to put it into these large windrows. They never started composting the way they should. The reason they put that material into the windrows was they could shrink that from, there's 100, get my other hand in here. There's the pile, and they could shrink that pile of organic matter down by 80%. So you have a lot more time to do things and keep putting waste materials into your landfill if you're reducing one of the components by 80%. But then it leaves people with this understanding that compost stinks very, very, very badly. When in any case, that in actual fact, it's not compost. If it stinks, you've got to manage it properly, and that's what we do.
Yeah, I've always, right. Anything that you do in your garden should smell like you want to smell it. And if it doesn't, then it's time to get rid of it.
Right. Don't put it
from
don't put it on anything you care about. You might put it on your weeds because
and
it
don't
kills.
Put it in your compost pile thinking it's going to fix it either, because you're going to ruin your compost pile.
Right. Yep. Unless you make sure that you are turning it on time. Well, but then every time you turn it, it slices and dices and crushes the good guy organisms in there. So you don't want to be turning compost very much at all. Just to the bare necessity.
We had an interesting little question, and we're going to take it on a little side note real quick. Do you forage for mushrooms, and what is your favorite things to go forage for when you do?
I am not very good at identifying really good mushrooms. I always go with somebody so they can point out to me the things that are good to eat, and then we'll head back to the house, and I'm happy to cook it for them. I'm just not. I don't trust myself.
But you do like to forage?
Oh yeah.
What's your favorites?
All of them. I don't pick them out. The only, what is it? I do have a bit of an allergy to. I'm not going to remember the name of the mushroom. They've got the really big mushroom body, and they usually put them in sandwiches instead of meat.
Portobello?
Portobello. Yeah. The first time I chowed into a hamburger with the portobello mushroom in there instead, it was like, wow, this is great. This is just so delicious. And about 2 hours later, I was just I spent the night kind of wrapped around the white fountain in the bathroom.
Oh, man.
Yeah, just was not. So I have to be really careful about what I eat.
Well, since we're running off on a tangent here, we got probably what is the most important question that you'll get asked all night long tonight. If you could be any animal in the world, which animal would it be?
I would probably be a river otter.
Oh,
I
I love
just,
otters.
yeah.
Um,
Yep, an otter is that's always, they seem to have such fun
yep,
in life,
that's what I
just
say.
constantly. They are, on top of each other, bouncing their waist. So, I'm panda. No, they're too big. Those river otters just seem so much fun. I can sit and watch them for hours.
I must have been having kind of a similar conversation with someone where I was talking about how they always look like they're having fun. And all of a sudden on Instagram, I start getting otter videos where over in, I think it looks like Japan, they like having river otters as pets in apartment buildings and stuff seems to be a pretty common thing. So you'll see people with, usually they'll have them in couples or pairs or whatever, and running around. And I'm just like, why can't this be my house right now? Like, so much fun.
Right? Why wasn't I raised in Japan?
Yes, yes.
Yeah, I would. Well, then, you would have thought that otters were, you know, too expensive to keep or whatever. So something would happen. Yeah, but that's my fave. It's sort of like, what's your favorite microorganism?
Okay, which one's that?
Well, I asked you. You've got to tell me.
Oh, mine?
Hmm.
I would go with protozoa, I guess.
They are cute. Any one specific type of protozoan?
I am not that knowledgeable to be able to get into a breakdown of them, but a protozoa are also nematodes. In Jeff Lowenfeld's, I think I can't remember which one of his book. He's got the picture of the nematode being strangled by a fungi.
Yeah.
I just absolutely love that picture.
Yeah, that's in his first book. Yeah, teeming with microbes.
Yeah. I'm working on his latest book, Teeming with Bacteria, right now. Got that sitting right here, and we're hoping to get him on that way sooner than later. I was in contact with him right before it came out, and he said he didn't want to do any more appearances until it had come out, and I didn't want to reach out to him until I'd read the book. So here it is. So once I get it done, we'll reach out and get him on.
Great.
Looks like
Well, he's gotten ahead of me by another book, so time for me to go find Teeming with Bacteria.
Yeah, and I got a question because I've been kind of nerding out on all your videos the last couple of days pretty heavily. So I was trying to remember what I should ask. Now, the one thing I remember hearing was like the plant exudates, they will basically ask for a certain nutrient. What is that interaction that happens that signals this, like, say, a request for nitrogen or a request for phosphorus? What is it? A chemical or like what happens?
It's a chemical, but it's an organic. So it's a mix of all the different amino acids that exist, and the carbon background that they get hooked to. So the plant realizes, and I'm going to be a little bit anthropomorphic here, so bear with me. The plant says, I'm lacking in boron, and gosh, I need a boatload of calcium as well. So that plant makes the exudate, that amino acid carbon mixture that's very specific, that basically, if you think of it as
a code of sort.
Yeah,
like
a code.
Like a one fit key sort of.
Yeah, what do they do back in the old days? The tap 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 Morse code. Yep. So it is an early Morse code, basically, with the mix of different amino acids on there is basically telling the bacteria and fungi that food resource comes in contact with to make this enzyme. And it's almost as if it forces the bacteria or the fungus to make that enzyme, and then the fungus or the bacterium go out a little bit further into the soil and release that enzyme into the soil, which now means the bacteria and the fungi can grab the boron and the calcium and maybe a few other things that the plant, multi tasked those bacteria and fungi. And so now all of the nutrient that the plant wanted is pulled inside the bacterium or the fungus. along comes a protozoan or a nematode or a microarthropod and chows down on those bacteria and fungi, and that's what releases the nutrient the plant asked for in a plant available form.
So safe to say, like to my friends that are even more layman than I am, a ratio of amino acids from the plant exudates will create an environment inducive to the production of a certain nutrient.
Well, it doesn't change the environment at all. It's a, you know, the root system of a plant is quite active, especially when it's growing, and so all it is
it seeks out that nutrient.
Yeah, it puts out that
Okay.
exudate. And I wouldn't call it a nutrient because it's just the amino acids and simple carbon structure, like with the code on the carbon structure to tell the bacterium or the fungus what it needs to do. And now they've got to go take those enzymes and pull it out of the sand, the silt, the clay, the rocks, the pebbles. That's where the source of all of that food is, that the material that the plant needs to grow and maintain good health. And it is the bacteria and fungi then that carry those exudates back to the root system. They have to get eaten by protozoa and nematodes now, and that's what releases the plant available nutrients. So it's like the bacteria and fungi are acting as the pantry, holding on to those nutrients until the plant requires it. Pretty cool way of
Pretty cool
doing
stuff,
things.
man.
Yeah.
And so the exudates are like their marching orders. The exudates come out. It's kind of like handing them a paper saying, "Hey, this is your job for the day. Go get it." And then it
Shopping list.
Yeah, shopping list. There you go.
That's it. Yeah.
The honeydew list.
Up on top, the next one, and the next one, and the next one. And when we get all of that pulled in, then we can go back to the root system of the plant and give our lives for, yep. So
Now is that how the cycling happens? Now does the bacteria kind of go over and sacrifice itself to the plant, or is it just more of like a digestion, or how does that work?
yeah, there's enough nutrient inside of the bacterium typically at that point that it can multiply several times. And so one of those bacteria presumably gives its life, but then there's been 100 or 200 bacteria replicating during that time. And so it's and watching, we've never really been able to label like a boron or like a calcium and look to see how rapidly that one particular molecule will go through this system. We can document the effect on nutrient concentrations in soil. That's the kind of data that we have, but we need to have it, or we don't really need to have it, but it would be fun to figure out exactly how fast all of these processes are going on in that root system.
And now, so each of these bacteria, these microbes and everything, is it, someone in chat was asking, are they like multitasking? Is it a specific bacteria for a specific element?
It appears to be multiple species of bacteria or fungi that can be influenced by the information coming from the plant. So the exudates from the plant are not that specific. There's, maybe 500,000.
Whoever can do the job can take the order.
Yep.
Kind of.
Okay. Yep. So you sort of I get the mental picture of the bacteria and fungi looking at the long list of all that. Well, let's see. I can't get that. I don't have that. I don't have that. Oh, here you go. Yeah, this is what we're gonna go do, and off they trot.
It's kind of like the chit line on a line in a restaurant. You got a bunch of guys standing there and they're all looking up which one's for my station. Quick to grab.
It's kind of like me looking at my dry amendments going, yep, you're going to get this one today.
And you will like it.
And then I had one more quick question. If you could pick one, with the dry amendment talk, if you could pick one thing that you would use as a dry amendment in, say, an indoor, if you've ever tried your hand at indoor tent growing before, what's one that's a good all around for,
It does.
general soil health?
Yeah, it depends on what crop you're going to be putting into that container. And so you have to have several factors that you're considering here. Is your soil just got bacteria? So what you've put into the bin is only bacteria in there and nothing else. Well, there aren't any plants that human beings like to eat that are going to need just bacteria alone. There's always a requirement for some fungi. So I would be, if what you were starting with was just bacterial, I would want to be putting in some compost that had a really good mix of all of the organisms. And then when you add that into your original compost, I would add in some pretty good fungal foods like humic acid. There are lots of humic acids out there because different kinds of the leonardite that the humic acid is extracted from. There's lots of different kinds of humic acid because if your leonardite is from the soft brown coal of a like an ash glade that has now decomposed to that level, or maybe this came from a conifer system, or maybe this area was oaks, or this was a grassland. You get a fair spectrum of food resources that the fungi in your system hopefully will utilize. So here's some of the testing that you need to do when you're trying to figure out what exactly is going to grow the fungi I've got in my biome, which fungi are going to be promoted. Well, so you tried this humic acid and it really didn't do much. Okay, so you know that you'll try that humic acid on something else. It wasn't what's going to bring the things back in this compost. So you try another humic acid, maybe a third one, and all of a sudden, boop, off it goes. Fungi just go crazy, and you get the balance of the fungi to the bacteria that you want. You have the predators in there helping you out by perhaps eating some of the too high level of bacteria. The fungi will help with bacteria, getting them into a stable condition, as well as fungi, as well as the whole community. So there's a lot of, you've got to think through this. You've got to understand the principles. And, there are just people that they're not interested. They want somebody to tell them how much of this, that, and the third thing do I have to go to the feed store and buy and put on my field. That's all they want to know. They don't want to know why that was supposed to work. They're going to end up with what they're going to end up with. But when we start using biology and you understand it, it becomes a very inexpensive process, because pretty soon you get all of those organisms growing in your soil, and you don't have to keep replenishing them year after year. Maybe you want to do a compost extract, or water once or twice with some of the compost that you've made, just to make sure you've gotten the system, the diversity back into the system.
So Dr. Elaine doesn't have a secret sauce that she's going to give up to G-Bay.
Oh, but I have that got me an interesting question though. What are your thoughts on like the Korean natural farming, the jadam, the things like that? When you want to add a little bit of pep to your instead of a compost tea or something, you make a JLF or something like that.
And I've seen those things not work at all well too many times. And I'll usually ask, did you look at this material with your microscope? What was in here? When you put that out on your beds, or you mixed it into the pond and now you're going to water with the pond water, they have no idea what the biology is that they put into the system. So maybe they added too much bacteria into a system that was already had too much bacteria, and you just make things worse. Sometimes you hit, by luck, by hook or crook, you manage to get a really good positive result. Well, okay, great. Now, why don't you take a look at um, that material and find out what the bacteria, fungi, protozoa, nematodes are in there, and then see if you can routinely get the same liquid that you're going to apply. But, too many times it's completely different biology because you made it separate from the first one that you made and you were highly successful. So I think they've got to do a lot. Well, maybe they need to do a lot more testing about exactly what they're doing, and they should be telling the people in their groups that they've got to be using that microscope to be able to tell exactly what they're doing. Are you getting, maybe this had, your compost had really good fungus in it, and now you've got another batch of the bokashi or whatever you're putting on, that was just chock full of yeasts, chock full of different kinds of fungi, and you took things way too fungal, and your vegetables are not going to like that at all. So
yeah, and that even tails into another one. We're all trying to collect, and we're learning these natural farming ways, and we love to collect microbes and throw them on our garden. Where is the point when we have added way too many of these things and we are doing way too much to our gardens?
yeah, you're working too hard. Um, get the biology up to the balance that your plants do very, very well. And a good thing to be measuring is plant sap. Um, we know what should be in the plant sap for many of our vegetables and grasses and uh, berries and orchard trees. We know what that plant sap should be at the different points in the growing season of that plant. And so why not be testing? You can just test your sap to make certain that your plant is getting all the nutrients at the highest level that it possibly can get. Great, you've got this down, and work out that pattern so you can do the same thing next year and the year after and year after. And the tool that you use is a microscope. Not that hard to run a microscope for an hour or two. My biggest problem with the microscope is I sit down at the microscope and I start looking at this stuff, and I'm starting to go, "Oh, look at this really cool guy over here," and forget to do the counting that's what I'm supposed to be doing. And I get too excited about seeing some critter I've never seen before.
Do you have a relatively cost-friendly microscope that you could recommend to people? Because I was just sending Fluff a message a week or two ago saying, "Oh, I want to get a microscope," and they're kind of telling the girlfriend the same thing. And I've heard you mention a few times that tonight and in other seminars you've done where $300-400 seems to get you one. That seems pretty decent. But which one, I guess, is the question.
The one that we're working with is LW. And they're in, oh gosh, I think they're somewhere in Southern California. Well, and they give a price reduction. If you say Soil Food Web School, that puts you right into you get I don't remember, I think it's like a 35% reduction in price of anything that they sell. And they are really good guys. They help a lot. If you're trying to put your microscope together, once you've got it, well, what's this piece for? You call them up and they turn their computer on. You turn your computer on, and you hold up the piece that you can't quite figure out where it goes, and they'll tell you, okay, you remember back when you put the headpiece on, you needed to put that in there, and they make sure that you get it in the right place and the right angle and all that stuff. It's, not really rocket science to put these guys together.
I think Fluff just breathed a sigh of relief because he's like, oh, that's one less phone call I'm going to get from this guy when he can't figure out how to do it. They'll do it, figure it for him.
Yeah, right. Yeah. Yep. That's one of that. They're really nice. They are very kind and very welcoming and of questions from all of us. If you have, yeah, there are a number of other microscope dealers that we have worked with in the past that were bent on making certain that we were very, very happy because we increased their sales. But you know, that somebody else buys the company from them, and they don't have any understanding that you know, this is not just a product that you can take home and stick it on the table and start looking through it. You need some training, and so that's why we encourage people to take the classes, so that you know the foundation course number one goes through all of the background information. I talk about all of the benefits that your plant can accrue from having the right kind of biology in the soil. So I've only touched on like half of the benefits so far. Maybe we'll get through to all of them.
What level of magnification are we looking at to start looking at these microbes and things?
We want to look at them in kind of different levels of magnification, especially when you're looking at it like a nematode or a microarthropod. You need either a 4x objective and a 10x eyepiece, or if you want to up the, you know, you really want to see the hairs on the legs of the microarthropod so you can identify what kind of microarthropod this is. Then you want to have a 10x objective along with the 10x eyepiece. So you've got a total mag of 100 there. And then you want to be able to get up close and personal so you can see the internal contents of the nematodes, and you can see the testate amoeba. Is that a test around its body, or is it just wandering around in an oblong fashion? Or to look for, you know, various particular things like with a nematode, does it have a spear in its mouth, and does it have knobs on the end or no knobs on the end? No knobs on the end means that's a fungal feeder. Knobs on the end says that's a root feeder. That you really don't want any of those in your system at all. So there we want a 40x objective along with that 10x eyepiece. And you really don't need any higher magnification than that. Sometimes you can get a little bit higher magnification if you get a camera that's very expensive. And so that's why I do not have one, because I can tell well enough that this is a flagellate, or this is an amoeba, this is a root feeding nematode, this is a bacterial feeding nematode, this is a fungal hypha. What kind of reproductive structures do you see in it? You don't need any higher magnification than that. A lot of our microscopes that we order from LW come with an oil immersion lens and it's just normal to have that high a magnification. We're not identifying bacteria. So we're just counting the bacteria. All we need to be is at a total mag of 400x. We do not need to go to 1,000 or 1,200x.
So if I say, for instance, I went out to the microscope store and bought one, came back, put it on my desk here, how would I prepare a sample? Is this like a slurry, a dry? Just go ahead and scoop some soil out of my pot?
If you want to be able to compare this sample with another sample with another sample, you better know how much of your soil went into the preparation. Now,
laboratory kind of stuff, like measured in milliliter, like, you know what I mean, or weighed out sample.
Yep.
Okay.
We try to make it really easy for the people, because you don't want to have to be, correct to the 1 nanoliter amount. And no, don't need to be that particular. So typically what we'll do with the solid is you get your sample from the field in cores. So you put 3 cores, and we usually go through a whole explanation of, do you need 3 replicates, or do you need 5 replicates, or do you really want 100 replicates or 2,000? And if you could go out and look at all 2,000 grams of your soil, you would know what's going on in your field very, very well. But you don't need to know that kind of detail. So 3 to 5 samples come in from the field, and each of those samples are 3 cores. You're going to mix that core material up in the plastic bag, get it nice and smooth if you possibly can. We're going to then take a test tube that is marked with 1 mil, 2 mils, 3 mil, on up to 15 mils, or whatever size test tube you buy. And then we usually go ahead and put 2 mils of water, and we can see that 2 mil mark on the side. And then we're going to start to add our sample into that water, and we bring it up to 3 mils, because it's volume that we want to compare. It's not air spaces, it's not number of other things. So now you've got you're up to the 3 mil mark, and you'll add the last 2 mils of water, and you got to make sure it's clean water. Every once in a while you want to look at what's coming through your sink and figure out that they just struck bottom of the aquifer. This is really not too drinkable.
The water's moving.
Yes, that's where all the extra bacteria came from. Goodness. So you do have to be a little bit aware of that. So now we've got a 1 to 5 dilution. We had 1 mil worth of volume of our soil, and we had 4 mils of water. So 1 mil of the sample divided by the total mils of the sample, and that's what you mean by dilution. So where things used to be concentrated, it's now been spread out 5 mils worth. You're going to shake that that water and your sample, and if you think of your arm going around in a circle, so around in a circle, we only want to go a quarter of the circle back again in 1 second. So I have my handy dandy little second minder here on my watch, and so I can go 1, 1,000, 2, 1,000, 3, 1,000, 4, 1,000 for 30 seconds. So you get it well shaken up. You don't want to take that sample and go, because you just destroyed 20, 30% of the organisms in that water, and you wanted to count them, not break them open. We want to get all of the larger aggregates broken up so that we'll be able to see all the things that were inside those aggregates. So that's designed to get the best extraction of the organisms. Once you're done, you'll usually set your test tube in a rack. You open up the top, you let it sit for about 10 seconds or thereabouts, so the bigger chunks of sand and things settle to the bottom. Your pipette puts 1 drop on the microscope slide, and then you put your pipette back, you grab your cover slip, you move that drop back and forth to get it well spread out, get those organisms spread evenly, and then you just drop that cover slip on that drop. None of that water should be outside of the cover slip. If you've got some of the material outside your cover slip, clean up that slide in the cover slip and try again. It does take a little bit of effort. You'll probably mess up for the first 3 or 4 sessions that you have.
I haven't done this since grade 10 biology. So.
Yep.
But
So
I'm interested. I'm getting the microscope for sure. I got this one here, but this is like a pest identifier. Amazon thing.
right. Yeah. That's something that Louis Pasteur would have used. So the father of modern microbiology. So
You got me thinking just a little bit. Are we all ruining like microbes and tearing things up and ruining stuff by shaking our stuff too much? Like when you just get a bottle of, say, any microbe product, and they all say shake well before opening, we shake the living shit out of it. Are we actually ruining our microbes at that point?
you're killing some of them, and they know that that death rate. So that if they're recommending shake like this, they know that you're going to be smushing things and just pancake and they're gone. They know you're going to do. They're going to lose. You're going to lose that many organisms, and so they account for that when they add the inoculum to that material.
Yeah, I would have never thought anything like that would have been that sensitive. I've never in my mind would I have thought that.
Now, in a lot of instances, if they actually have spores, those spores are a lot more rugged. You can do a lot more things if it's just the spores in the sample. And like when they're selling you something like Bacillus thuringiensis, they sell those as spores. And there you don't have to worry. Drive over it with a logging truck, not going to harm them. So if they're live living organisms, you have to treat them gently. So yeah, work accordingly.
I think sometimes we forget they're alive living organisms because we can't see them, and we're big and dumb.
Yeah, well, you start to appreciate them more as you've stared through the microscope and you start learning their names and what it is that they do. And so all I've got for G-Bay, is that your name? Did you get all of your questions answered? Yep, okay. I was just worried that we left our organism.
No, I've got to take time to absorb that. I can't take any more right now.
Okay.
Thank you very much. Thank you.
What's your thoughts on using things like biochar?
You got to check and make sure that there's a need for the biochar. We were working in real sandy soil when I was at the Rodale Institute in Pennsylvania. I was the chief scientist for 3 years with that group. And one of the things that we did was to add biochar in different concentrations and look to see what the benefit was. Well, the field that we happened to choose was it was almost a sand dune. There wasn't any organic matter in there. There wasn't much life in there, but we added the different levels of the biochar to that field. And then we had a crop, I believe, of soybean in one of the fields and a crop of corn in the other field. And at the end of the growing season, we took samples of each of those higher levels of each level of the biochar or no biochar at all. And there was absolutely no difference. Because it's sand. Sand does a real good job of infiltration when you already have decent levels of organisms. They weren't high, but they were decent. And so there was no benefit of the biochar in that particular experiment. Whereas if you go to a soil that's really compacted, so remember when we were talking about, the compaction layers like this and the root systems, and so they'll all go sideways. When you add biochar in after breaking up that compaction layer, now the last time you go through, put the biochar in there. And in those instances, we see a lot of benefit of putting in a biochar because you need to have something that's going to leave the air passageways, leave the openings in that soil, until the bacteria and fungi, protozoa, and nematodes can get down there into that soil and rebuild using the normal ways of rebuilding soil. You've got to have good organic matter. It's like, um, if you can't, if you are working with something that doesn't have an organic matter level greater than 3%, you're not going to be able to keep your microorganisms alive and happy. There's not enough extra food for those organisms to stay alive. So you might have to do biochar to keep the airways and the passageways open, but that's that's only in certain kinds of instances do we have to have that in order to see the benefit of the biochar.
So if we were to just take that to a small scale, little guy doing a no-till bed, say, a 4x8 section. So, he's built the soil. It should have plenty of organic matter in it. Are you going to see the benefit from that biochar in there? Is that really where you're going to see the benefit of it?
Yeah, where you have a soil that you can break up, say for the last time ever, you're going to till. And you're going to put in some organic matter. You're going to put the biochar in there, which is organic matter. Now you'll probably be successful there. Whereas if you're dealing with sand that's already kind of friable, it's not really compacted at all, you don't see much difference. And then there's all the in between those two endpoints that, how much sand and silt and clay, when does the clay become something that you've got to put biochar in order
That
to...
is exactly what I was just going to ask, because I'm kind of where our soil is more clay based, typically where I'm at. So, it might be a good thing to add to my soil in that matter.
Yep. And so, I'm interested in doing the additional work to be able to say, here's the threshold where you really do need to start putting in biochar, or you can do some tillage for the last time ever and then once you get all those microorganisms into that system, they build the structure in the soil. Well, that's what you're trying to do with the biochar as well. You're just trying to provide enough openings for these organisms to live in, and then they build the structure, and that's what prevents the compaction from happening again.
Excellent. Someone was asking earlier, and that's a good one for me. I liked it too because I use it. Neem. What are your thoughts on that? Do you use it? Any issues?
I have always been very disappointed in neem because it doesn't really seem to do the job until you start getting it fairly concentrated, at which point it becomes a toxin. You kill the organisms in the soil. So there's just so many questions that I have around that. How did they make the neem? Exactly what was their process? Did the neem get burnt? Did it get too concentrated? Is it actually glommed on to some toxic chemical that was, too many? There's a whole research life to for.
How about using like the meal in your soil as just like the neem meal from the processing thing that they make into like a fertilizer? How do you like that in the soil?
Again, it's a case of how toxic is this, and there's some, we aren't getting consistent responses. So until they want to tell me what they've done in making the neem in a liquid form or in ground up with the other stuff, if they can explain to me what all they did, then I would be able to do a much better job of figuring out why this killed everything and why things grew rather nicely. So it's when someone's trying to protect their intellectual property, they're not likely to tell you what exactly it is that they did. So, my recommendation to them is start using that microscope and figuring out what you're really doing.
Yeah. I think I found the same results you have. If I use it in a concentration for, let's say, if I'm foliaring it, if I use it in the concentration to actually kill something, then I always see weird things happening. And I think, like you say, toxicity, whatever. But when I've used it just kind of as a foliar feed, just kind of spray it on just as a very light preventative, it has always seemed to actually be quite nice to the plants. They've responded very well to it, but definitely used with a very light hand.
Yep. And so where is that threshold? How much can you actually, and do you get incrementally better kill of the disease causing fungi or the insect pests? When do you go over the edge? And that's what we've got to figure out. And then when how was the plant material picked? How was it treated? How long has it been in a completely dry condition? In which case the efficacy is going to go way down if you completely dry that material.
Yeah, kind of breaks those oils down that are supposed to be being used, right?
Yep. And
Okay.
if the oils aren't in there anymore, I don't think we're going to get much effect.
Right. That makes total sense. That to me seems like then that would be more of something you would likely use as a process it down, use it as the top dress instead, or a soil building structure rather than to make a spray out of. So yeah, that makes total sense.
Yep. And then again, it's use your microscope to determine whether things are improving in your soil and it just hasn't improved enough for your eyeballs to be able to see it, or you can see that everything's going south. And what are we going to put in there to combat the neem oil?
You're absolutely a pioneer. Done a lot of groundbreaking work in soil microbiology. I'm kind of curious though about the future. What do you have any future projects going on that you can talk about, or like where would you like to see things head in terms of soil research and making the world a better place?
Well, I think we need to convert all agriculture over to a biological approach. We've got the basics in all over the world. We need to have people going to the different parts of the world and making compost in order to sell to local people. So, you want to stay within your biome. You could stick your operation right on the, here's a biome out here. There's a different one over here. There's a different one here. So you could have quite a large facility. But I really think we have to stop trying to kill Mother Nature, and we've got to start treating nature with respect. We don't want to be killing everything in the soil. We want to make certain that any management we do is promoting the beneficial organisms for the plant that you want to grow. And what people don't seem to be picking up on is the fact that as you go through the various stages of succession, you're typically starting out with no plants growing in the system after there's been a devastating catastrophe happening. You're back down to ground zero. The big forest fire, or on the eastern side of the Cascades in California, here in Oregon and Washington. Those are just devastating fires. I've never seen anything like that before, and I've lived in this part of the world quite a bit. I don't remember as a child ever having those kinds of unstoppable flames and quite so often in so many acres. So things have definitely changed. Um, and so we've got to, realize catastrophic. That's the first stage of succession. And you've got to have the weeds. You have to have the undesirable things to start growing in that highly disturbed, very impacted, well, it's not soil, it's dirt. There is no biology typically left in the dirt after a catastrophic event. So now you've got to start building the soil once again. And the first step is strictly bacteria. And they are the first thing that comes back. The photosynthetic bacteria come back. You get the true bacteria starting, and you start to build that plant community once again. As soon as some part um, of that plant material, weeds though it may be, as soon as that cellulose is now present in that next stage of succession, now you can get fungi growing in that soil. And now you can start to grow herbs, and you can start to grow vegetables. And typically the things that we're looking at is the brassica community, very early successional kinds of grasses, not highly productive. You don't have to have 3% organic matter in the soil at that time. Things are just getting started, but pretty soon more plant species come in. You get more kinds of carbon molecules in those plants, and now we get more fungi growing, and we can start growing cabbage and onions and garlic, and start moving into better, more productive grass systems. Once we get to an equal biomass of fungi and bacteria, we can grow the most productive grass species, the ones that we want to have growing in our fields, because if the highest nutrient concentration can occur there, we're going to be saving people from a lot of starvation. You don't have to eat a lot of different things, but you need high material, high nutrients in that material. So we're going through this successional process. As we go over the edge into we are now fungal dominated, now we've set the stage, or Mother Nature set the stage for growing shrubs and bushes, and on into um, um, forest systems. Deciduous forest systems usually come first, and then into the conifer forests, and then into the true old growth species. So there's that successional process. Where do you want to be? If you've got much more bacterial, you're going to be growing these plants. If you've got a right on 1-to-1 ratio of bacteria and fungi, you're growing these plants successfully. They are what are getting most of the nutrients. They're the ones that are highly useful for human beings to be consuming. Move into orchards and into conifer systems. What is it that you want? And the biology is a little bit different. So there's not one size fits all. You've got to think about what you actually wanted. And so we can't ever go back to dumb farmers. You've got to be thinking about what you're doing. You've got to have an understanding of what's going to be the least expensive and the most beneficial. We work with people all over the world where we can show that when they start getting the biology back on their soil and are building the structure coming along, that where we're matching the understory species to be the same as the crop that we're trying to grow, then we stop having to do so much work. And in the first growing season, when you compare the biological approach to the chemical approach, we don't use any inorganic fertilizers. So take that bill and kick it out the door. You don't need that inorganic fertilizer anymore. There's no reason, unless you're really trying to advertise to everybody that you are a farmer that really doesn't know how to grow things. We also want to note that in those places, we don't put out any pesticides. We don't need to put out those materials that kill stuff. And typically, if you are backing off on your amount of fertilizer and on the pesticides and the trying to kill the pesticides, the bad guys, if you will, you have saved typically in a 2,000 acre farm, you've saved over $3 million. How would you like to put $3 million back in your pocket, never spend it? Could you do some good elsewhere? And so we've got to get people understanding that this is well worth their time to invest in farming, because we can grow much higher concentration nutrients in our plants, much higher sap, plant sap is going to be produced. The exudates that fall on the surface of the soil are going to build that soil faster. And within a year or two, you can basically put one application of the compost tea out on your agricultural field. And typically that's put down right where you're putting the seed down. If you're dealing with annuals, if you're dealing with perennials, you just go out and apply compost extract as a liquid. And that's all you have to do for your farm until you come and harvest it in the fall. Was that a convoluted and a roundabout way to get that point across?
We don't mind roundabout at all around here. You should see some of the tangents that we've gone off out of nowhere from time to time. So no, this is great. Just absolutely loving this. It's once again, thank you so much for your time.
Well, it's my favorite thing to talk about. Have you noticed?
Yeah.
We may not look like it, but we're kind of, we're kind of fanboying out right now. I am, anyways.
I had a question with winter. You talked about disasters happening and things like that. What happened to these microbes in, like, Cariboo and I both live in a place, it was minus 48 just before Christmas here. So what happens to those microbes in the soil when it's that cold outside? And what are things that we might be able to do to prevent them from, maybe perishing in the cold or whatever, I guess, degrading in any way?
Because we are trying to choose the local indigenous species, the things that survive that wintertime temperature are going to be the ones that they're in your
so they're meant to be there, so that's their zone.
Yep. And there are a lot of bacterial species that grow better when they're down in the negative values.
Okay.
So there's usually an aggregate that they're inside. They're in the middle of the aggregate, and it's like I kind of think of them as they're all huddled down there, keeping each other warm.
Yeah, like the hens in the coop outside.
Yep. So they have dormant stages, almost all of them that live in biological, with the biology lives with the plant. They all have dormant stages. And so some part of the population will make it through. The most rapid rates of decomposition that have ever been measured on this planet were from soil in the middle of the wintertime, protected by about 6 inches of snow. So down below the snow, the water was at 4 degrees Celsius. Sorry, I'm thinking in Celsius, but what is that? That's like 34 degrees Fahrenheit. So the soil was frozen right on top, but underneath that, because the ice freezes and it takes the coldness away, you have a layer of bacteria and fungi, protozoa, that are happily working away, doing their jobs of decomposing, and the most rapid rates of decomposition ever measured on this planet are found there, like on
Wow.
mountainsides. So when we first reported this result in ecosystems and the environment, um, there was a kind of an outcry that, oh, you're crazy, or you don't know what you're talking about. This is just, no one else has ever reported on this. And it spurred several other research groups to go out and repeat what we did. And so like 3 years later, all these reports came out about, well, we went out and we checked how rapidly things decomposed in the litter bags, and we have to come to the same conclusion. They had Hunt, Coleman, Ingham, and there were one or two other people probably on the list of credits, the authors. We have to come to the same conclusion. The most rapid rates of decomposition occur in the winter under the snow. Which
So would
is,
that also be the same? Would that carry over to your compost pile as well in the winter?
yep,
Okay.
yep. Typically, the middle of your compost pile doesn't ever freeze.
Right. Yeah.
It's got good organisms in there, and if it does freeze solid all the way through, don't worry. When it thaws, they all wake back up
Go
again.
back to what they're doing.
hmm
Right on.
Probably at a diminished level, but we're not seeing any loss of species.
Okay.
We don't have the money to
Mhm.
test absolutely everything that we're working on for DNA analysis. Woof. Because you start looking at natural, real soil that grows plants all on its own without any help from human beings. Um, uh, there's so many different species, like TG, Dr. TG at the Center for um, Microbiology at um, let's see, it's one of the Michigan schools. He said that in a woodlot, that like what is a typical woodlot is 25 paces by 25 paces by 25 paces. Um, in a woodlot, there was more than 1 million species of bacteria in that woodlot. And then you go away from Michigan State University, and you go away from that first woodlot, and you look at a second woodlot, and yeah, it may only have 1 million species of bacteria in it, but they're different than the first. And when you start thinking about, and that was just the forest, the woodlot. What would happen if you were in an old growth? What would happen if you were in a extremely healthy uh, agricultural field? What would it be? What would the diversity be if you were in a riparian zone, or if you were in a swamp, or if you were, just think of the diversity of, that's just bacteria.
Mhm.
And fungi are probably right up there with it. Maybe, maybe not 1 million species, maybe only, 750,000 fungal species. And we just, every time somebody who really does good work in the taxonomic field, when they go out and they start looking at all these different systems, they keep finding new things. We have not even begun to touch the diversity of what's out there in the real world. So there's a lot to learn. There's a lot of promise in what we're doing. If you've ever read any of Dr. David Johnson's publications, it was probably about a year ago that he um, said that as we increase the fungal biomass in these different agricultural systems, the amount of carbon being sequestered went right along with the amount of fungal biomass. So we could sequester tons of carbon dioxide equivalents in a growing season in a 1-acre field. And now he's gone back and deliberately grown ever increasing amounts of fungi as well as bacteria, because he kind of thought that, you know, bacteria do pretty well. There's a lot of bacteria. There's got to be a lot of carbon in there. No, there's no gotta be. Don't tell Mother Nature what she is or is not doing. Um, the fungi, because he got even higher levels of fungi than he had before. When you look at that fungal biomass and the amount of carbon that it is sequestering, he estimated, and this was in his paper that he presented just a couple weeks ago. I won't remember the name. I'll try to track down the name for you. I know my assistant will be able to tell me that. But what he said was that given that we can get the organic matter into the soil, that we can get these microorganisms growing rapidly in the soil, building structure, getting root systems down to the probable distance. You know, if you've got 10 feet of plant material above ground, you've probably got 10 feet of roots below ground. If you're a plant, you know, think of the sequoias, think of the redwoods. How tall are they? 300 feet? Well, then the root system probably goes down 300 feet. So if we're sequestering in all of these places, we could very reasonably sequester all the carbon dioxide that's been released into the atmosphere, and we could put that carbon back into the soil in about 6 months. We could solve climate change that rapidly if we started to demand that people listen to and worked with nature instead of against nature.
It always makes me laugh when some human somewhere thinks they can make a new system. I'm like, I'm not the smartest guy out there, but I can see that the world outside the way it is has been working for long enough, and she's got her dialed in. We don't need to be. We can play with it, but don't try to improve it or be better than it. You're gonna
Well,
fail.
how can you improve something that you don't know how it works?
That's it. You know what I mean?
So at the very least, we've got to start understanding how all of this works, how all of it works together. And then maybe we can have a little conversation with Mother Nature and say, could we tweak this just a little bit? What do you think will happen? If you do that,
Absolutely.
you're going to blow up the planet. So don't do it.
We had a little question here in chat, and it is kind of an interesting one. Like, when we transplant plants, a lot of us like to use our inoculants, or myceliums, stuff like that. Whatever that we inoculate with. Is there anything specific? How do you feel about those types of products? Are they worth using? All those things, in my thoughts, all those things should be in my soil already, and I shouldn't feel like it needs a pre-inoculation to get ready to be where it's going to be in home.
Yep, and that's what we're trying to do with the compost, making it properly so we maintain all of the diversity within that soil, and we just all the time keep increasing the local indigenous species. If you get a jug of microorganisms from, let's say, where do you live?
Actually, that's funny. I was going to bring this up a second ago because I buy a specific organ. When I don't make my own, I buy one that is only about 3 hours away from me.
Okay.
So I'm trying to keep it as local as possible. But yeah, I do that exact thing, and I was going to ask you about that.
But it's like I run into people that, they ask me this question, and then my response is, well, where are you buying these jugs of microorganisms? And the answer is something like, well, I live in Southern California, and I've been buying these inocula from the people up in Pennsylvania. And I kind of have to look at her like that and say, okay, so what's the likelihood that something that was born and raised and bred in Pennsylvania, and think about that outside air conditions, the amount of humidity, the temperatures. Are any of those organisms going to survive even a week or a month if you put them out into a basically semi-arid to arid grassland in Southern California? Those organisms are just going to belly up and die. You just paid a lot of money to put a little bit of organic matter back into your soil. That's all it's worth. So look for the local indigenous ones. Well, where are you going to find them? Well, in your compost. How do you increase the diversity of organisms in your compost? Go out and find a different starting material. And you only need a small amount, a handful. And you mix that into your compost pile. And if that set of microorganisms can find compatible places to hang out, that's where you'll find them. And they'll be available for that exudate coming out of the root system of your plant. They'll be available to grab that and go, woohoo, look at this. The chick only wants me to bring back really good stuff anyway, and then they're going to pay us even more. So not a bad system. Mother Nature is very clever in the way she runs things. So I would say don't waste your money on things that can't survive in your soil.
Right.
And how do you know that they're not there anymore? Microscope. Get your microscope out and you cannot find it anymore. It's probably not there.
Yeah.
See, this is how I justify that microscope purchase to the girlfriend right there. As Dr. Elaine says so. Everything in this conversation just keeps coming back to microscope. Look and see for yourself. I'm like, well, it really seems unavoidable at this point.
Yep.
If we all do it together, we'll take the punishment together, boys. Okay, we all in this together.
Here we go.
Next week, we're all buying brand new microscopes.
Yeah.
They can't all divorce us.
Yeah.
I'm moving in with you.
Well, I'll be looking forward to seeing your faces in the foundation courses. So we go through all of the different benefits that the biology in the soil will give your plant. And then we talk about how do you manage the compost pile right so that you're going to be able to get rid of the bad guys and maintain the good guys. And then we do the same thing for extracts and teas. And then we get real deep down into the microscope.
Nice. Did I see that you were running kind of like a little special right now for signing up on your thing to get some of the foundation courses for free, correct?
Yep. Not a bad deal.
Out there, get on it. That's right. Free education never hurt nobody.
Get it while it getting's good. So it will only be another couple weeks, and we will be back to the normal fee for the classes. Only $5,000. But
only,
in the
right?
only.
Let's get in on that deal, folks. To anybody that isn't unaware, we already put that link in the description for this before the show started. So go click that up, and it should take you right to the website. So, Dr. Elaine, we will literally sit here all night long if you let us sit here with you. Are you kind of on any kind of time constraints, or you want to wrap her up? I know it's getting to be about 9 o'clock ish where I'm at.
Well, maybe just take a few more questions because I do. I have I have a lecture that I've got to get ready that I give on Saturday in Virginia.
Okay. Wow. Okay. I got one that, I've been, in terms of composting, I've heard from quite a few people that say, avoid using manures. And one thing I keep hearing you saying is, just get rid of the bad guys. So, and I've also been told it is doable to use manures, but you really got to kind of watch what you're doing because they're a little more dangerous. Do you have any advice or tips on that? Or is there any manures that we should definitely avoid, some better than others, or?
Yeah, you want to take a look at the what the animals are being fed. So if their food is just chock full of toxic chemicals, well, we want to give them this antibiotic. Well, okay, you're putting something toxic into your soil, into your compost pile by doing that. And so what's the probability that you're going to be able to grow the whole diversity of the good guys if you're trying to kill off something bad that, you know, there's no chemical that has effects on just one species of organism. If you're trying to take out, E. coli, you're also going to take out most of the enteric organisms that are beneficial for your digestive system. So now you're dealing with a less than complete set of organisms to do the job in your soil. So you want to avoid using those toxic materials. Please realize that most of the inorganic fertilizers that you put out are going to be salts. And we often think of just table salt as being salt. That's just one type of salt. It's table salt, sodium chloride. And when that sodium chloride gets into the water, or it gets into a liquid form, as in around the organisms in your soil, that sodium and the chloride will disconnect and try to hold on to the hydroxide and hydrogen of the water. It takes water away from your plant. It takes water away from the microorganisms. The microorganisms don't have a lot of reserve. The plant can have a lot of reserve, but a bacterium, does it have much water in it? And so instantly you add a salt into the soil and you're killing bunches of these organisms, the good guys as well as the bad guys. But you got to have the good guys for all of these things to work, for all of these things to function.
Say you knew exactly what your inputs were for the animals. Would that change the answer a little bit?
It might, because if you look at cow manure, are the animals being routinely dealt with? They're routinely getting some antibiotics, or they're routinely being washed down with something. You want to start to encourage that person to leave those things behind. They probably don't need it. If you could get some of their manure that was clean, didn't have any toxic chemicals in it, then you could make a really good compost, bring that back, and the animals, the cows, you could lower the toxic chemicals lower and lower and lower. Now, with horses, for example, they don't usually give the horses constant antibiotics or constant materials. It's just like once every month or once every 2 months, depending on where you are in the world. They're going to give the horses deworming medicine, and that's a real killer. You want to, from the second they started applying the deworming material, you do not want to touch that manure. Go put it in a big pile and let other things take care of. That is going to be a 2-year process to get that manure back into where
usable.
Usable
Don't
form.
some farmers actually use like labs to help with the gut worms in like their cows or things like that? Or is it the labs that helps with that? I know they do something with the labs to help with their gut, but I was just wondering if you would ever have a problem. I mean, that would never be a problem, correct?
I would need to know exactly what the material was that they're adding. Is it purely organic? Is it normal materials? So, and then think about the fact also that when we're building our compost piles, we only need 10% of the volume of that compost pile to be high nitrogen. So it's not massive amounts of manure that you're trying to go find. And you can always use legumes. So, if you've grown peas and beans in your garden, when you harvest the peas and the beans, and you've got all that residue material still standing there, cut it, and there's your high nitrogen for making your next pile. Alfalfa. Why not grow a field of alfalfa? And the only thing there that you want to look for to make sure that the alfalfa is actually fixing nitrogen, increasing the concentration of nitrogen in the body of the plant, is that when you pull up the root system and you see the little nodules, the little round nodules, you want to cut that in half and look inside, and it should be the color of a drop of blood. It should look just like a drop of blood sitting there on that cut surface. If it's only kind of pinkish here and there, you're not getting much nitrogen fixation. If you've got nice, bright, red colored liquid, that's real good.
I'm so glad you said that. I've never thought to look at that. I've known about the nodules, but I did not know that you could cut them open, check the color, and wow, okay.
Yeah, really easy, so why weren't you told this information as farmers? So there's so many little tricks like that that we convey as we're doing the foundation classes. I don't even think about them anymore. Well, of course you're going to use these kinds of approaches. But so, often when we're starting at a new place, we'll 60% woody materials, so like dried leaves from that fell to the ground the preceding year. If you harvested hay and you let it sit too long out in the field and all the nitrogen is blown off into the atmosphere, you really got straw. Well, that's good woody stuff. So 60% of your pile is made from wood chips and things like that, that I just went over. You want 30% of the pile being green plant material. So recently cut, or as soon as you cut it, you got it dried very rapidly. All that nitrogen, all the nutrient is still intact inside of that material. And so you can use that as green material. And then 10% high nitrogen. So, when you're putting that pile together and you think you've got really good high nitrogen material that you put into the pile and you keep taking the temperature every day and it's just sitting there, it's a 50 or, it's 80 degrees, it's 85 degrees the next day, it's 90 degrees, and then it's 90 degrees, and then it's 90 degrees. And you know that the high nitrogen wasn't party food. The green material isn't doing what it's supposed to do. So in that particular instance, we want that compost pile to work. So we'll go get blood meal and put that into the middle of the pile. And you'll learn pretty rapidly, how quickly rapid growth of the bacteria and fungi is going to take that temperature up. Just be a little on the low side with the blood meal when you're trying to figure this out, figure your pile out. So we do a lot of work with you when you're taking the foundation courses of how do you know when you've got enough? When should you stop? What are the other ways at using? If the temperature is now at 180 degrees and you know that things are starting to go anaerobic because the microorganisms are growing so fast in that pile, you can't keep up with them. You couldn't turn that pile fast enough to shut down the bacteria and fungi. What do? Yep, that's called chimneys. We will put chimneys. So you're going to have to come and listen to the foundation classes so you know what I'm talking about when I say chimney.
I had a pile start smoking on me once when someone told me how to increase my activation with blood meal, and I was like, all right, well, if a little works, a lot will work better. And nope. Yep, I'm pretty certain I was about a second away from it actually igniting.
Oh, I've had moments like that too. Yeah. So we all live and learn. So yeah, just the first pile that you make sure that you're out in kind of an open area. So in case.
It's good to know.
Yeah, but that thermometer is a real important piece of equipment when you're doing composting, because you don't want to get anywhere close to 180 degrees. That's just too scary. So then you learn how to make really good compost. And it usually takes, 3 piles for people to really get it and start getting to have a good feel at these starting materials that you're putting into the system. So always be asking, like if you go and get hay, if you get straw, you want to ask that grower, uh, what exactly did they do with this plant material? Did they apply glyphosate? Did they put on 2,4-D? Did they, what were the toxic chemicals that they put on? What was the fertilizer that they used? What was, um, the, like you noticed there were a lot of Colorado potato beetles. Did they treat the hay with something to kill off the beetles? And if there's just a boatload of different kinds of toxic materials, you've got to turn around and leave because your pile's not going to function normal. So pretty quickly get to the point where you are making your own starting materials. Works just fine for most folks. Like I collect the dead leaves in the fall, and every time we mow, I put the grass into, I get it dried and put it in plastic bags, things like that. So I'm not having to go spend money to make my compost with.
I like that. Trying to close the loop on it. It's great.
Yep. It's if all you're buying is a small, like 4 pounds of blood meal, and that's all you're spending to grow 3 or 4 good piles of compost, you can handle that. It's when you've got to go out and pay, $200 for the woody material and another $100 for the green plant material. That's where this doesn't really um, compute all that well until you've got the um, recognition from from your neighbors that you make really good compost. And how do they find out about it? They look in your garden, they go, whoa, look at your tomatoes. Why don't my tomatoes look like your? Well, because I had real compost in here. You want to take some home with you and put it out on your tomatoes and see if your tomatoes will spring into action. And so help them out a little bit so you
I
start.
tried keeping up with my neighbor's garden, and I was unsuccessful. I also found out that his plot was for 30 years a hog farm, and now he's growing tomatoes on it. Yeah, like 20 years after the fact, and there's no way I can keep up with that.
Yeah. So watch your neighbors carefully and see which ones are going to benefit from this. It's like back when we first started and we got people working with us. We'd bring them the compost and they'd spread it out. That's great. That's wonderful. And then we'd come back in a couple of days and want to put the thermometer in because it was enough. Enough amount of compost that it should have been still heating a little bit, and all of everything in their bed was at room temperature or at ambient temperature. There was no heating, and it was like, what did you do here, guys? Um, oh, we didn't do anything. We followed all your instructions, just exactly. Um, I'll come back out here with a spray and I'll spray with a compost extract because this is not functioning the way it's expected to. And so, come back and apply the extract and get things going. And then you come back, a week later and everything is back to ambient temperature. And I realized finally that I should be coming on a different day and see what they're doing on their spray day. And they were spraying glyphosate on that bed. And why are you doing that? Don't you know that the glyphosate is a sterilant? It kills everything, including you. Oh no, no, if there was anything like that, they'd tell us. Why? So the glyphosate going onto that material just killed the organisms. And so the temperature didn't rise. It didn't do what it was supposed to do. We didn't control the diseases. Well, you just turn around and leave. You've got to just say, look, when you finally have decided to follow instructions, let me know. I'll be back then, but not until.
Does it? I'm imagining it doesn't take much of the glyphosate to actually stop a compost pile from being very effective, right?
Yeah, doesn't.
Okay. Yeah, I was just thinking. Last year, I hate the fact that they spray a fence line somewhat near my house, and I did notice that at a couple sections throughout the summer that all of a sudden my compost pile was a little. It wasn't working.
Yeah, a
And
little wonky. Yep.
yeah, a little wonky. And even though I couldn't figure it out, I don't have my microscope, but I can't figure that out. But the only thing I could nail that down to is it always happened somewhat near that spraying time. So I imagine even just a little bit of overspray, just
drift.
really.
Yeah, and we need to have the legal system finally get hold of, and um, you know if they kill all of the organisms in the soil on your side of the fence, they should have to pay to bring those organisms back. So I'd like to, yeah, when we're finally making boatloads of money, we'll hire a lawyer and get them going on this. So it's just not right that the guys with the toxic chemicals are killing things on our property. But
Destruction of personal property or private property, whatever you want to call it, right? It
Yep.
very well could be looked at. We've got a question in the chat here asking if you've ever done any work with microbial insecticides like fungi or microbial isolations.
Sure. Yeah, quite a bit, especially back when I was at like Texas A&M and at Colorado State. We were much more interested in them because we thought we would need to put those kinds of organisms back into the compost piles, back into the soil in some way, because they would have been destroyed by all of this the tillage in the soil, and the applications of inorganic fertilizers and pesticides would have killed off all of those guys. That was our reasoning. And we discovered that really didn't hold water. Those good guy organisms are out there. If they're normal in your systems, they're still hiding out someplace where human beings haven't been out there applying toxic chemicals. So just develop the maximum diversity that you possibly can. And if in doubt, let's say you never see an organism like one of those real beneficial organisms. Sometimes it's a fungi, sometimes it's bacteria, can be protozoa as well. And make certain that you've got lots of good taxicabs, which are the higher level predators. Typically, they will get all covered with this sets of bacteria and fungi, protozoa, and nematodes, and everywhere that they walk for the next couple days, they're spreading that inoculum every place they go. So dung beetles, got to have those if we're in a grassland. And you can always add those guys as eggs, or you can put them in when they're very young. And as long as you maintain the biology, so the bacteria and fungi are growing just fine, you can probably bet that all of those other organisms are going to be able to survive.
So to take something like, say, a compost tea or a worm leachate tea and dilute it down and use it as a foliar, would that actually work as sort of a pest deterrent and kind of an all around just general do everything for your plant?
Yeah, most of the time when we're looking at you know having the materials on the outside of the plants, on the leaves, on the branches, on the blossoms, on the fruit, that's really triggered within the body of the plant the trees, the plant itself has to have adequate nutrients present, and it's got to have the balances of those nutrients present. And the only way to get more of those nutrients into the bodies of the plants really is to get nutrient cycling in the root system going again. So some plants need, triple the amount of boron. Other plants will need only less than the normal amount of iron or zinc or so. And all of that balancing of all those different nutrients is done really by the plant putting out these messages of to the bacteria and fungi in its root system to bring me back the nutrient I need. And as long as your plant has those proper balances, the plant's going to be producing those chemicals that shut down the pests and the problem organisms when they move in for an attack. So it's not like we have to put in these special things. We got to find the right bacteria. You should have the right bacteria. And you've just got to make certain that the plant's at full metabolic nutrition. It's just one of those things that comes along with the territory.
Yeah, I think we all kind of we take this and we run with it. And we think we need to just throw it at it nonstop all the time, as maximum levels as possible, and so it does make it a little.
You need to. What are the signs? What's the information that? And if you're watching closely through the growing season, it really only takes a single growing season to start figuring out all of this out. Every summer is a little bit different. Have any of you ever noticed in your lifetime this summer was exactly like that summer back 10 years ago? Nah, it's always different. So
I wish I could just plan on which crop to grow that year. Like if I could just get that part right, I'd be happy.
Is it going to be a drought year? Or is it going to be so wet we don't? Yeah, I mean, I look
that's all I want.
It's just tell me whether I should be, keeping all the water in or letting it drain through.
That's right.
And that's one thing of getting rid of all of the compaction in the soil. You will finally have the proper kind of infiltration moving water into the soil and then keeping enough of that water so that your crop plants can make it through that dry season.
Right. We had somebody in chat asking about your thoughts on bricks. That's kind of basically what you were talking about earlier when talking about testing the sap in the plant, correct?
Yep.
So you find that very important to test the bricks on the plants to see the nutrient uptake, how everything's doing down there.
Yep. It's a bit different because the plant sap is usually from a much larger, closer to the ground, and you want to break open that sap material and send it into the lab for that analysis. Whereas with bricks, you're really looking at I'll remember. It's
bricks is like the sugars in the plant, correct?
It's one of the components of the what's in the and I'm just not coming up with the name. I love it. This is what happens when you're 70 years old. You forget words every once in a while. So it's the percent dissolved carbon. And sugar, of course, is one of the highest percentages of that material. But you want to be looking at both things. So when you put your drop of sap on the little window, close the lid so that the sap that you've got is evenly spread all over the window. And then you hold that up to the light. You can see the total amounts of sugars or other compounds. There's another compound or two that's in there. I'm just not going to remember. And then with the amount of sugar, you want or amino acid, sorry, that's amino acid, you will see that fuzzy line. If it's really fuzzy, you've got a lot of amino acids in there. Whereas the sugar is the top is white and the bottom is gray. So you can get that kind of information and kind of follow along and how's the plant doing, but recognize that you need the same outside conditions to be the same from day to day. So if you're going to go out and start taking bricks, do you want to go first thing in the morning? No, because the plant has not pulled in all of the sunlight it needs to have returned to equilibrium. So noontime, for example, is a good time to take your bricks readings. But what if it's now clouded over? If it's a cloudy day, your plant's not making as much sugar. It's not going to be doing, it's not going to be building as much amino acid. And so you might be surprised by the lowness of a value every once in a while if you take it when you're in the middle of a cloudy day. So try to make sure that it's always bright sunshine. You're not in the shade. All these, try to make things just as consistent as possible. So you know that the reason why the sample number was low was because of some toxic chemical was sprayed on it and it's not functioning very well at all until it gets rid of that toxic chemical.
I have a funny feeling that LW is going to see a big rush on microscopes getting sold here in the extreme near future. Are you able to throw like a good book out there for identification purposes for all the microorganisms that we'll be seeing under these microscopes, so we can at least say that is a this is a that, and this is that kind of a nematode, etc.
We go through all of that in the foundation course.
Yes, great answer.
Yeah.
Great answer.
So, and we try to get to that pretty rapidly. So in the opening lecture, I go through all of the organisms and talk about all the identifying characteristics. When we get to the last course, the foundation course, we go through lots of different kinds of materials. And if you can, you'll have different organisms because you'll be doing your work at your house and there'll be some different critters. And so when you can't identify something, we ask that you send a picture. And so you can have a real fancy camera set up with your, if you've got the 2 eyepieces and a tube for putting your camera on, that makes life a whole lot easier. But I took all of my pictures just using a camera and snapping pictures through the eyepieces. I have very steady hands. If you have hands that shake, okay, you're not going to be able to do that. You're going to want to buy a pretty good camera. And we often have ongoing discussions about which camera is best. And so we do that all the time in the different ages. So we have a forum for the foundation classes for people to ask questions. We have a forum for the people doing the advanced microscopy. And then if you want to become a consultant, there's another group and we help people go through the whole process of converting dirt back into soil. And there we make certain that it's you that is making all the decisions of what to do. We will have been telling you lots of information all the way along, and now you've got to prove that you were paying attention.
I like it. I like it a lot. I think that seems like a pretty good spot to wrap her up and let you get on with your night. I like sincerely from the bottom of my heart, thank you so much. This has been a lot of fun. Hopefully later on down the road we can get you back for a part 2 at some point in time.
Absolutely. Yep. Well, I was late coming on, so I sort of feel like going a little extra overtime was only fair.
Well,
I want to thank you as well too while I have a little chance here. Thank you again so much.
Yep. Anytime.
Yeah, I'd like to take a second to give you thanks. This must have been one of the most informative, educational episodes we've had here on the Cariboo Hart TV. Thank you very much for taking time from your busy schedule and sharing the wealth of knowledge that you have tonight. Thank you.
Yep, it's my favorite thing to talk about. So just invite me back anytime.
Hey, we're gonna hold you to that.
Okay, I'll be expecting it.
Absolutely, and I want to say the exact same. Your work, it's invaluable. Thank you for everything you do, and I can't wait. And I hope we get to talk to you again soon. Thank you so much.
Yep. Well, keep up the good work.
And you'll see my face in the foundation courses for sure. We're starting a farm this year, so I'm going to start with your course to get us on track. So
Okay, great. All right. Well, you have a good rest of the evening, and hopefully talk to you soon. See you in the foundation courses.
Good luck in
awesome.
your travels.
Thank you.
Yep. Take care. Ciao.
Thank you.
All right. Oh, and just like that, she's gone. Okay. I thought,
She's fucking good at that, man.
okay. Well, you know what? That means we need a dab.
Yeah.
We need a dab. This one's for you, Dr. Elaine. Thank you so much. Oh, wow. That was awesome.
Hey, Cariboo, thank you for getting real uncomfortable and reaching out and doing all the legwork to make that happen. I kind of just threw this to you just as a whim because I really just wanted to talk to Dr. Elaine, and you made this shit happen. But thank you so much.
Yeah, well, that was wow.